So the next speaker is Carolina Muri from NIH. She's going to talk about P47.
I'm sorry, how do we...
Oh.
Okay, thank you. So first I would like to start by thanking the organizers for the opportunity to tell you about our work. We are interested in developing a transmission blocking vaccine that targets PFS47. Sorry, I tried to use the laser and I went by. So PFS47 is expressed on the surface of female gametes and ookinase. And this gene, we know, we started working on it because we know it allows the parasite to evade the mosquito immune system by inducing damage with a silent cell death, so the mosquito immune system is not triggered, and then the parasite survives. We think this is one of the reasons why malaria transmission of Plasmodium falciparum malaria is so effective. There's also evidence that this protein is required for optimal fertilization of the female gamete. So in order to develop a transmission blocking vaccine, the first thing you have to do is to express the antigen, and this protein has 14 cysteines, so that's not trivial. So after 2 years of trying all sorts of things, we ended up with a fusion of thyroidoxine, and we expressed the fullest protein in E. coli. And at this point, we started the immunizations, and there were good news and bad news. The good news is that we developed, the protein was immunogenic, developed good antibodies. The immunofluorescence was positive. Here is P47 that stains both male and female gametocytes, but in the mature gametes, it's the female that expresses this protein mostly, and here we use P230 as reference. So this was good. And then the bad news came. None of these antibodies, we did 14 monoclonals and many polyclonals, none of them had any significant transmission blocking activity. So after 3 years into the project, we were ready to drop it. And we say, okay, if we bother to make these monoclonals, let's see where they're binding. So we start doing that. And then when we did this, there was a surprise. We found that 9 of them bound to domain 1. This protein has 3 domains, 1, 2, and 3. And then the other ones to domain 3, none of them bound to domain 2. And we have genetic evidence that that's the key domain for the function of this protein. So, okay, so then it's obvious we have to then express domain 2. But that's easier said than done because domain 2 has been baptized as the domain from hell. Any cell you put this domain in, it kills the cell. You cannot express it, at least not in vivo, maybe in vitro systems. So in a moment of desperation, we went and we replaced these 2 cysteines for alanines to break this loop that is predicted. Sorry, I didn't want to have the sound effect. So when we did that, then that removes all the toxicity. You can make milligrams of this protein in any cell, and it's fine. So, okay, so we went ahead, we immunized, and we got really good transmission blocking. Our immunizations, everything I'm going to show you, were done with 1 prime and 2 boosts because we were screening. And then we used between 200 and 300 micrograms per ml of purified IgG in everything I'm going to show. So it works in Anopheles gambiae. It doesn't want to move forward. Okay. It works in Anopheles stephensi 2. And then when we did this, then we also first expressed the whole domain 2, and then we did a series of deletion and monoclonal antibodies, and we mapped that the transmission blocking is given by this 52 amino acid region immediately upstream of this loop. And just as an anecdote, if you make antibodies to this linker region, antibodies to that region actually enhance transmission. So we removed that region from our antigen, and we're left with this 52 amino acid region. So we went ahead, and then we wanted to see how this vaccine is working. So we first wanted to know if it will be enhanced by complement, like 230 is, and we find that in the presence or absence of human complement, the vaccine still works, so it's not enhanced by complement. In Stephen's it's the same thing. So we conclude that this vaccine does not require participation from the human complement. Then, because we are mosquito people, we wanted to make sure that how this antibody was working. Could it be that it was now the mosquito immune system that was killing this parasite? And this turned out not to be the case. Whether you have an active complement system, this is with the intact immune system in these 2 controls, or if you silence the final effector of mosquito complement, it still works. So it does not require the mosquito complement system either. Okay, so then we went ahead and Alvaro started counting how many old kinases form, and there is a substantial reduction in the number of old kinases. We also looked at the expression of old kinase specific transcripts, and there's also a dramatic reduction. So based on this, we conclude that most likely these antibodies are working by blocking fertilization because it's in the surface of female gamete. Okay, so this is, I hope I convinced you that you have a nice antigen that is promising, but then how do you move from these to a vaccine candidate that really should move forward into clinical trials and so forth? So what I have learned is all about potency, so basically how good is this antigen so that at physiological antibody levels and as time goes by and the antibody levels go down, how low can these antibodies go and still protect people? So I think this is a major determinant. So our antigen is linear and is small. It's only, the final one we're working with is actually 58 amino acids. We had a few extra amino acids. So it can benefit from conjugation. So we've done that. We have worked with two systems. One is the AP205. I think it's a very easy to use system, relatively speaking, and very effective. So we have worked with this, and we have already generated this protein. We have started the immunizations, but I cannot tell you the results yet because the mouse are still working for us making the antibody. We have also we've been working with Patrick, and he, together with Scaria, they have done the conjugation, classic conjugation with exoprotein A. And we already have immunized. We've done the second boost. So next week we'll be collecting serum, and I can tell you, how much better is this antibody. Also, all our immunizations were done with a commercial antibody called Magic Mouse, which is a CpG-based adjuvant that we know is not ideal. There are stronger ones, so we can go with GLA, LSQ, or ASO1 type adjuvants. But also, we think it's important to optimize the route of immunization. And I want to share some data with you about this. So this is a classical, in our initial studies, we were doing intradermal immunizations, and when we did a single boost, usually we got about 70% reduction in transmission using 200 micrograms per ml. So we went ahead, and now we were decided to try the intramuscular route. And we had a big surprise. We were using half the amount of IgG, and we got 100% protection. And we have never seen something like this. So we went ahead and we titrated this antibody. And when we do that, what we find is that we get full protection up to 25 micrograms per ml. This is the protein without conjugation and without the optimal adjuvant. So our hope is that by optimizing these parameters, we hope to move in this range, because I think that will put this as one of the very promising vaccine candidates, because it can generate very high transmission blocking at low antibody titers that you will have, you can sustain in a person over a long period of time. Hello? Sorry, this is. The other question is, do people have natural immunity to this protein? So we first did a screening with serums that were provided by Pete Crompton, and they're from Mali. And about 12% of people have antibodies. For young children, it's about 10%, goes up to about 20%. However, the antibody titers are low. Just in comparison with MA1, you can detect them, but they're low levels of antibodies. We also, Peter also gave us serum before and after the transmission season, and this is to the protected region of P47, and people do have antibodies that increase. So this tells us that there might be some natural boost. We think that the other important parameter to keep in mind is, can we make a universal vaccine? How much variation are we going to have in the region? And we find that there are 7 haplotypes, and haplotype, I mean any amino acid difference between the sequences. Everything I showed you was done with haplotype 1, and all the testing was done with, which is the most common in Africa. All the testing was done with NF54, which is haplotype 2, and clearly those two cross-react. These two together make about 85% of what is found in Africa. There's other minor haplotypes, and then in Asia, haplotype 6 is the most dominant one. And now we are expressing all these different haplotypes, and we're going to see the affinity binding of antibodies to HAP1 to these different variants. And we also have a transgenic that expresses this haplotype, and we're going to see if the antibodies still block transmission with equal efficiency. So we think that there is good prospects that this could be a very broad vaccine. Also in the New World, in the Americans, only haplotype 1 is present. So we think this is a promising vaccine. And the bottom line is that we started with the idea that to develop transmission blocking vaccine, I understood you have to take the full length protein, immunize with everything, should be positive to the immunofluorescence on the parasite surface, and we found that yes, when you express the full length, you make great antibodies to this region. All these antibodies are useless. The region that you need antibodies to is hidden, and then you really have to work to find it. And we hope that by targeting this region, then we can find a weak point in the transmission cycle that will allow us to prevent infection and hopefully prevent human disease. And I just would like to finish by introducing the people who did this work. The vaccine work was led by Gaspar Canepa, working very closely with Álvaro Molina Cruz, and they also received help from Lampourville, a graduate student in the lab, and Adi developed all the AP205 viral particle work. And she's now a graduate student at CSU. So with that, I would like to finish. Thank you for your attention. This work was done at NIH. Thank you.
Thank you, that was great. You mentioned that the upstream linking region, antibodies against that, enhance transmission.
Yes.
Do you have any hypothesis for what the mechanism of that would be?
So I have hypotheses, but it's just theoretical. It's what I imagine, right?
Mm-hmm.
Everything seems to indicate that this protein is not static, but is very dynamic. If you saw the IFAs, it's mostly intracellular, but there are patches where it's translocated to the surface. And we think it probably undergoes some conformational change and then interacts, I assume, with a male receptor if it's involved in fertility and is in the female gamete. So what we think is that the monoclonal that binds the linker region may stabilize this open conformation and optimize the fertilization. That's our theory, right? But in practice, we don't know. That antibody has very high affinity binding and has a strong effect. Actually, if we put the domain 2 complete, I didn't say it here because it was so confusing, but when you immunize with this linker included, you start with, after one boost, you have great transmission blocking. If you do one more boost, you lose the transmission blocking because it all goes to the bad region. So we had the good, the bad, and the loop. That was our, the antibody from hell had it in its domains. So we trimmed everything, and now we're left with the good part. So we removed the troublesome loop, and then we remove the bad epical, and now we consistently get only good transmission block.
Thank you.
Hi, Caroline. Thanks. Very nice. A few things emerge. One is the knockout parasites are very fertile, right? In
Yeah.
falciparum. So that puzzles me, of course. And do you have an explanation for that?
Yeah. So the parasites do, this is something that's been puzzled us for quite a while. In Barrillas, it's clear that P47 has a fertilization phenotype, meaning you don't make, in vitro you cannot make organic cultures in Barrillas. What we think is happening is that in falciparum, it's not essential, but it's required for optimal fertilization. So if you have a parasite that you put thousands of gametes and you give it plenty of time to fertilize, some of them will make it, right? But I cannot mention the details, but I know someone has been doing some genetic analysis. If you have another strain that is wild type, the wild type wins by far. So if you do a cross with a wild type and a knockout, the wild type P47 will win. So I said in the lab, it's like, when you have a boyfriend, right? If there's no one else to choose from, even the ugly one will find a partner.
Yeah, I'm
right?
still with you, yeah. The other question I have is the potency of the monoclonal. I think
Yes.
You didn't show that. You saw 25 milligrams of IgG after immunization. Well, what is the monoclonal doing in terms of potency?
Yes, so what happens is we only have one monoclonal that has strong transmission blocking, but is not optimal. The affinity binding is much less than the bad monoclonal that enhances. So now that we know exactly the target region, we are going to do the next set of monoclonals. So basically the monoclonal we have gives good good transmission blocking, but the affinity of binding is not very high. At
Can
least
you give me a
penalized
figure?
is less.
Can you give me a figure?
Excuse me?
How many micrograms do you need? Or
I think that with that monoclonal, Alvaro may correct me, but I think we can get up to 50 micrograms per ml. But with a polyclonal, we can go to 25. And our benchmark, I don't know if I'm in the right bulk, but we want to go below 10 micrograms per ml. I think if we get there, we can say with confidence that this is a very promising target. Right. But I think that the monoclonal depends on how many you have to choose from and how good you are when you screen and get the really best one. We have many, but many of them were unstable, so we only have one that has been very stable, and that, unfortunately, was not the one with the highest affinity. We have a collaboration with Bob Cedar and Pete Crompton on which we are taking serum from those patients that have antibodies to this region, and we're doing, trying to get B cells from people from Mali to try to get natural human high affinity binding antibodies to this target
but your
region.
prevalence of anti-47 is pretty high in these Malian people, right?
Excuse me?
12% had anti-47 antibodies? Which I
Yes,
thought
but
was
this
huge.
is, the 12% is to the whole protein, right? To the target domain, it may be less. I mean, the antibody titers are less to the protective region. And the other thing, they have detectable antibodies, but they are low, right? Those IFAs, those ELISAs were without 1 to 100 dilution. So you can detect the antibodies, but they are not at the levels, I think, that will confer transmission block. They're low.
Do I have time for a question?
Yes, we can.
Thank you for a really nice talk. Rupa Baines from Nature Communications. I'm sorry if I missed this in your talk, but how often would you need to administer or re-administer sort of this vaccine?
Yeah. So,
and the second part of the question is, how would that work in non-endemic or low transmission areas?
So first, for our screening, when we're trying to find any region that will confer protection, we did a prime and 2 boosts. And our assays were done between 200 and 300 micrograms per ml. Now, once we define the key region, so now we're optimizing, right? So now we're only doing a prime boost, because it is in a normal situation, that's what you would do. So what I show with the intramuscular injection is just prime boost with the 58 amino acid peptide without optimization and without conjugation. So we don't know. I presume that the antibody will protect similarly into low and high transmission. Now, where it may play a role is, I don't know how strong the boosting effect, natural boosting effect may be, and that may make a difference, but I really cannot speculate of how important that boosting could be. In an ideal world, my dream is that we'll end up with a microneedle patch so that immunization will be as easy as to put a Band-Aid on a child. End of the transmission season, you pull the first Band-Aid before the rains begin, second Band-Aid, and they cover them for the season. That's our dream. But of course, it's fantascience at the moment.
Thank you.
Hi, Adrian Hill, Oxford. Very nice talk. Maybe I missed it. You showed us the structure of the intact protein with 3 domains, and domain 2 is the relevant one.
Sorry, that's a model. So this is a model. The structure has not been solved because domain 2 is super flexible, so we cannot crystallize this protein. Now, we know we can model very well domains 1 and 3, and we know that they interact with each other. Domain 2, we cannot visualize it because it's very loose and it moves, so we cannot.
So does it work as a linear 58-mer peptide?
Yes, the peptide is linear, yes. Meaning, we take the peptide, it goes into inclusion bodies, and then we wash it with soap, then put it through urea, and then we remove the urea, and then we get a stable, soluble, pure peptide. And there's no disulfide bridges. So at least we don't have to do, I don't know what conformation it takes once it's by itself, but we don't have to do anything. So we think it's linear.
And the one monoclonal that you have that recognizes domain 2,
yes.
Have you tried to map within domain 2 where that binds?
We know, but we have mapped it to this 58 amino acid region, we know, but we have not done the detailed mapping. Meaning amino acid, we have 58 amino acids, we have not done the precise epitope mapping.
Okay, thank you.
If someone is interested in doing that mapping, we'd love to send you the monoclonal if you could tell us where it is, because we don't do that kind of thing. Thank you.
Thank you very much, Carolina.